Okay, I think we can go ahead and get started. Hi, everyone. Really excited to introduce Elena. Elena Simon is a PhD student at Stanford in James Dow's lab. Her research focuses on reverse engineering machine learning models of proteins to extract biological insights and advance model interpretability. She has previously worked on a range of computational biology problems, including cancer bioinformatics, protein language models, and small molecule drug design. So I'll turn it over to you.
Yeah, thanks for the introduction. So, as Vicky mentioned, I'm Alana Simon, and I also have to say I've watched so many of these seminar talks on YouTube, so it's cool to actually give the talk this time. So, today I'm excited to share some recent work where we've been applying mechanistic interpretability techniques to understand how protein language models actually work under the hood. And feel free to interrupt with questions during the talk in the chat, or happy to take any at the end. We should have some time there. So before we dive into reverse engineering protein representations, I want to talk about why we even want to do that. There's certainly the intellectual appeal of reverse engineering these models to understand how they work. But beyond that, I think this research has actual implications for model development, deployment, and potentially even biological discovery. So first, let's walk through how kind of our approach to studying proteins has evolved over time. So traditionally, understanding a protein's structure or function meant extensive lab work, protein purification, modifications, crystallization studies, functional assays. And while this approach has built up our entire foundation of protein knowledge, it's inherently very time-consuming, low throughput, and many proteins are also just particularly challenging to study this way. So the field took a huge leap forward with the introduction of fast sequence alignment and structure prediction methods, where these computational approaches let us take things we've learned from well-studied proteins and extend those insights to new proteins based on their sequence and structural similarities. Now, the beauty of this approach is that we only need a protein's sequence, which is something we can much more easily obtain. But the catch is that we're still largely limited to finding patterns that we already know about, and our predictions tend to be most reliable when we're working with proteins similar to ones that we've studied extensively in the lab. One particularly promising computational approach has been the development of protein language models, or PLMs. These work just like language models that predict words in a sentence, but instead predict amino acids in a protein sequence. So here we see a protein language model that is trained via masked language modeling, where it's trained to predict masked out amino acids in a sequence. And what's fascinating is that even though these models aren't given any prior knowledge about structural or functional annotations, they're only trained on a sequence prediction task of predicting what amino acid goes into what position. It actually seems like they learn many of these properties entirely on their own. And this isn't just speculation. We see evidence of this learning in how these models perform. So researchers have successfully used PLMs to design stable functional proteins, and their internal representations can be used to predict structural and functional properties way better than just looking at the amino acid sequence alone. And these embeddings even help us identify evolutionary relationships between different proteins. Now, when you think about it, this isn't that surprising. To correctly predict what amino acid belongs in a specific position, the model needs to understand that position's context, essentially the local environment this amino acid exists in. And this environment is fundamentally shaped by the protein's structure and function. Plus, we know that functionally important positions in proteins tend to be conserved across evolution. And since these models are trained on many evolutionary variants of the same sequence, they can pick up on these patterns. Now, to actually investigate these hypotheses about what PLMs do or don't understand, researchers have looked to their successes and limitations, both across different tasks and then also across different proteins. So looking at per task performance, we've seen clear evidence that PLMs can capture both 3D structure and coevolutionary statistics between positions, so what positions kind of evolve together over time. However, as we discussed actually in one of last year's talks, while improving the PLM quality leads to better structure prediction, not all downstream tasks see the same level of improvement. And so by examining which tasks do or don't benefit from improved PLM pre-training, we can try to refine our understanding of what information these latent representations may or may not contain. And then we can also learn a lot by looking at which specific proteins the models struggle with. So for instance, proteins with fewer neighbors in the training data tend to perform worse, and protein isoforms created by alternative splicing, where the model's predictions sometimes get nonsensical outputs that can only really be explained by realizing that the model is actually just predicting the structure of the protein that would make sense for the most common protein form, and then incorrectly applying it to all other variants. And these two together kind of both suggest that PLMs might be heavily relying on recognizing conserved patterns in proteins from the training data rather than learning fundamental physical principles. But the thing is, so far, we can only really make educated guesses about what PLMs have learned based on these successes and failures. But what if we could actually look inside the models and more directly examine what they've learned? Even if they are just memorizing patterns from the training data and applying these to new sequences, rather than learning some generalizable energy function over all protein sequences and structures, there's still so much we can learn from investigating whatever it is that these models have memorized and how they use that information. So this brings us to our central questions. Can we actually decode what PLMs are learning? Can we understand their internal representations and algorithms for modeling proteins? And if we actually can understand how PLMs process proteins, what can this teach us about biology itself? So we already have promising signs that this kind of interpretation is possible. Researchers have shown that by examining attention patterns in transformer-based PLMs, we can predict 3D contacts and identify binding sites, which again, if you think about it, isn't entirely surprising. After all, an amino acid's identity naturally depends on the residues it's close to in 3D space or those it interacts with at binding sites. But what's crucial here is that the model was never explicitly taught about these interactions. It was just trying to reduce loss on its masked language modeling task, and in order to succeed at that task, it discovered these patterns on its own. And more importantly, we can extract and interpret this information. Now, finding 3D contacts might not seem revolutionary given tools like AlphaFold exist, but imagine this. If we could identify a model's mechanism for detecting a specific type of binding site, we could potentially discover new instances of that binding site that just haven't yet been annotated. Or even more intriguingly, we might uncover mechanisms that identify entirely new types of functional sites that haven't been characterized. Or perhaps we'll find that PLMs use completely different algorithms than humans do to identify binding sites or allosteric interactions. And then what might we learn from these alternative approaches? So from this kind of scientific discovery perspective, trying to understand what PLMs learn could open entirely new paths to understanding protein function. But this could also transform how we approach model development. So imagine that instead of just observing which evaluation metrics go up or down when we modify architectures or training data, we can make more informed decisions based on like a specific understanding of how each change to our model actually affects the model's internal mechanisms. So as you might be familiar with, the architectures used for protein ML these days are so complicated. And while many design choices within these models can be motivated by introducing inductive biases into the model, at the end of the day, these architectural choices mostly come down to what makes the loss go down, even if we don't really know why. And it'd be super helpful when designing models to have a more informative lens into what exactly each of these changes in the model does beyond just these loss ablations. And then finally, how these models process proteins can actually impact how the users interact with them. So if we can really more concretely understand their failure modes and limitations, that'll impact how users interact with them. And potentially, if we really understand their internal processing, we could even intervene in this processing to guide our outputs in customized ways. So to pursue these goals, we use techniques from mechanistic interpretability, where this field generally aims to understand neural networks at the level of their internal computations, essentially trying to reverse engineer how they process information. So looking at this neural network diagram, each little neuron in the network performs basic mathematical operations, typically matrix multiplication followed by some nonlinear transformation. And mechanistic interpretability focuses on understanding what these individual computations represent and how they work together to kind of compose the model's behavior. And by studying these internal mechanisms systematically, we can start to piece together how different parts of the model work together through these circuits of neurons or little subgraphs to process and reason about our inputs or proteins in this case. Now, when this type of approach has been applied to language and image models, people have found many neurons that activate on consistent semantic concepts and then work together to form fairly human-interpretable computational subgraphs of neurons within the model called circuits. So here we see three car-related neurons working together to form a car detector, where you can kind of identify what a neuron activates on by seeing what inputs lead to a non-zero value as the output of this neuron. However, most neurons activate on multiple, often totally unrelated concepts, such as the legs of a cat and the front of a car in this particular example. And this phenomena is often called polysemanticity. One effective way to address this is by taking the embedding vector for each token, whether that's a word or an individual amino acid, and training an autoencoder to transform it into a much larger and more sparse vector. So typically, the autoencoders you might be familiar with learn to reconstruct your input from a hidden latent representation that's usually much smaller than your input, which forces the model to learn how to compress your input into just the key, most critical pieces of information, somewhat similar to PCA. However, with a sparse autoencoder, we don't want to compress our data, we actually want to expand it. This is based on the assumption that our polysemantic neurons, also known as the hidden dimensions of our embedding vector, are actually a linear combination of truly interpretable features that just exist in a different latent space with a much higher dimension. And if we can just train a model to properly transform our data into this new space, maybe we can just analyze features in their expanded interpretable form. And so this approach is a form of something called dictionary learning, where we can think of our autoencoder as learning a dictionary of vectors that can be linearly combined to reconstruct our input. So in this framing, the ith row of our autoencoder, specifically the decoder in our autoencoder, would be considered the dictionary vector, specifying the direction in the original latent space that the ith feature represents. And as these dictionary vectors are the learned weights of the model, they're the same for all input. And then each input varies in terms of how much of each dictionary vector or these features it contains. So when you pass an embedding vector through your autoencoder, the value in the ith position of our new expanded large latent vector is the feature activation value, telling you how much the ith feature is present in your current example. Visualized differently, each embedding vector x can be represented by some bias vector plus a linear combination of learned dictionary vectors where the coefficients for how much each vector contributes to this particular embedding is specified by these feature activation values. Now, you might find it a bit counterintuitive to assume that our complicated embedding vectors that are just secretly linear combinations of simple interpretable concepts. And this is definitely a big assumption and most likely doesn't capture the whole story, but it's building off something called the linear representation hypothesis, where we've actually seen some evidence for this over the years in various domains. And even if it's an approximation, it can be a pretty powerful approximation. So some of the first evidence of this linear representation hypothesis came from observations in the early world of word embedding models. So these models, which learn latent vectors to represent words, reveal surprisingly linear relationships between concepts in this high-dimensional space. So a classic example expresses this pretty beautifully. So if you perform something called vector arithmetic, where you just subtract the embedding of the word king from kings and apply that same vector to queen, you'll get queens. And so this suggests that the concept of plurality exists as some consistent direction in this embedding space. And then similar patterns emerge for the concept of gender or tense or other linguistic features. And so you might imagine that the most intuitive way to encode these linear relationships would be to dedicate each dimension in this word embedding space to a specific semantic concept, essentially creating a unique basis vector for each possible meaning. Under this scheme, a dimension would activate or have a positive value when its concept is present in a word and then remain inactive or have zero value otherwise. But to actually include all possible semantic concepts like this would require an enormous number of dimensions, most of which would be zero most of the time. So just from an information theory perspective, this is highly inefficient. Moreover, neural networks aren't explicitly trained to represent data in this sparse interpretable representation. And so indeed, research has shown that models do not represent things this way, and instead they naturally learn to encode these concepts much more efficiently by using vectors that are slightly non-orthogonal. So rather than representing each concept in its own, unique perpendicular direction in this latent space, the model compresses information by allowing concepts to share dimension through kind of subtle overlaps. This achieves similar levels of expressiveness while dramatically reducing the required dimensionality of our vectors, and also is what leads to the polysemanticity we observe in our representations. So this is where SAEs, sparse autoencoders, come in. So the purpose is to unpack these compressed overlapping representations into a sparse space where each hidden dimension actually does correspond to a single interpretable concept. So they essentially reverse the efficient but kind of opaque encoding that's used to reveal the underlying semantic structure of our data. So when we apply these sparse autoencoders to language models, people have found remarkably clean and interpretable features. So for instance, here Anthropic discovered that their cloud models contain a feature that activates specifically on content related to the Golden Gate Bridge. And fascinatingly, this holds up across multiple languages and even on images of the bridge. And so to demonstrate the causal nature of these features within the model, you can synthetically activate this Golden Gate Bridge feature across arbitrary inputs, and you find that the model consistently steers its responses to just incorporate the bridge into every question, regardless of what the original question was about. So in this case, you can see someone is trying to ask about 20 questions, and the model is just making the answer about Golden Gate Bridge. Similar results have been found when asking about baking recipes or any question you are interested in, the answer will be about the bridge. And so while this particular steering example is very entertaining and wholesome, it also illustrates something profound about these features, which is that even though this linear representation hypothesis may not be entirely perfect, decomposing our representations into linear combinations of concepts still reveals features with precise interpretable effects on the model's computation. And the fact that we can use these features to reliably influence the model predictions also just opens up exciting possibilities for better understanding and controlling the model behavior. And so we wanted to ask whether this approach would work on protein language models, and could we use these techniques to try to understand what PLMs have learned? Now, while protein language models share core architectural elements with natural language models, both being generally built on transformers, it wasn't immediately clear whether the same interpretation techniques would transfer. Unlike the Golden Gate Bridge example, where both feature activation and steering results are intuitively interpretable, understanding what the analogous features would look like for proteins has its own unique challenges. So we had a few fundamental questions we wanted to answer here. First, are the neurons in PLMs also polysemantic, even given their smaller vocabulary and just the fundamentally different conceptual requirements in modeling proteins versus language? And can we extract human interpretable features using SAEs, or do these models encode protein information in ways that are just too alien to our human natural understanding? And then what's the best approach to even interpreting these features? Can we develop scalable, usable methods, or just require a lot of time-intensive expert analysis? Do these features have clear causal roles in the model that we can use for steering the outputs? And crucially, can we extract meaningful scientific insights from these features? After all, one of our primary goals is to try to learn new biology from these models in the first place. So today we'll talk through how we approached each of these questions and what we discovered. So this shows the overall pipeline in which we run a protein sequence through a PLM, in this case ESM-2. Then from a single layer, we extract embeddings for each amino acid, run them through a sparse autoencoder, and extract the learned features to analyze instead of looking at the individual dimensions of the original neurons. So when training an SAE, we ideally want our features to be as interpretable as possible and also capture as much of the original model as possible. However, we can't explicitly optimize for either of those. So instead, we optimize for our features to be maximally sparse under the assumption that this will increase interpretability, while still being able to accurately reconstruct our original embeddings. We do this by measuring the similarity between our reconstructions and the original model embeddings, and then also applying an L1 loss to the features themselves to encourage the features to be sparse. Now, if we didn't incentivize the features to be sparse, it would just be trivial to reconstruct the embeddings, as you could just do two identity transformations. But on the other hand, if the features are too sparse, you might not actually capture everything in the embeddings, and this might not be an accurate representation of the original model representation. So we then evaluate our models according to these two criteria, and we can also measure our reconstruction quality by taking our reconstructions, putting them back into the PLM, and seeing how much this now impacts our downstream masked language modeling prediction loss. As if these new reconstructions really do capture all of the key information in the model, your final predictions will be just as good as the original embeddings were. Now, there are a bunch of ways people try to improve our ability to optimize these two different losses that mostly come down to variance in the nonlinearities used after the encoder in your sparse autoencoder, which are also a critical piece of trying to incentivize interpretability in your features, and a few other small chaining details, but they all generally do the same thing. So this is kind of a reasonable overall visualization of how this works. So here, we're looking at the activation patterns of four different features as highlighted by the proteins that maximally activate them. So to get a quick sense of what a feature is identifying, you can look at the top, N proteins that most strongly activate this feature. And you can also look at the proteins that activate this feature along the whole spectrum between zero activation and maximal activation to try to understand what is it generally picking up on. And so here we visualize each feature by highlighting the activated amino acids and various proteins in pink. And you might notice that the patterns and distributions of amino acids that are activated varies a little bit between the different features, but also there's something clearly consistent and seemingly interpretable in what each of these four features is identifying, both in terms of which proteins are maximally activated and what positions within those proteins are being identified. So in language, people often ask how often a given feature is activated as one way to kind of characterize that feature. But here we can get even more specific in how we characterize the feature activation patterns before even trying to understand what these features mean. So we can break down the frequency of an activation into first, how many proteins activate a feature at all, and then second, when a feature does activate some kind of protein, is it just looking at one amino acid or a full domain? And by looking at proteins at kind of different quadrants of the spectrum, you'll identify totally different types of features that have different activation distributions within them. And then you can even go a step further and ask, when a feature activates on multiple amino acids per protein, what is the relationship between these different amino acids? Are they sequentially clustered together? Are they structurally clustered together? We find both. So just as we visualize the activation pattern along the structure of the protein, it's also helpful to visualize it along the sequence of the protein. And depending on the feature, you might care more about one versus the other. So on the left for each of these features, we see a protein sequence described by the single letter code of each amino acid, where the height of each of these tiny little letters corresponds to the feature's activation value for that amino acid. And the kind of colorful little lines at the bottom are just all of the non-activated amino acids squished together. And combining these two visualizations helps highlight some of the differences between some of these features that activate in structural clusters or sequential clusters or a combination. So it looks like there's something specific the model is highlighting in each of these features. And if you spend a while in time all looking at the proteins and reading up on their functions and their different domains, as I kind of had to at the beginning of this project, you can build up some intuition for many of the features, but there are thousands of potential features to analyze, and similarly, thousands of protein properties that these could be identifying. And since the bioinformatics field has already put so much effort into characterizing annotated proteins, can we just leverage that? One way is by comparing the feature activation patterns to known protein annotations. Many groups, like in this case, the Swiss Institute of Bioinformatics, have curated wonderful databases, like SwissProt, of functional and structural annotations. So can we check if these annotations are the types of features and concepts learned by the model's SAE features? So for each SAE feature, we want to check whether its activation is predictive of an existing annotation for any kind of biological concept, whether it's a structural pattern, a binding site, anything we have data on. So we effectively want to learn a classifier that tells us whether above a certain level of activation, this feature's activation is predictive of the presence or absence of these biological concept annotations. And as we're comparing thousands of features to hundreds of concepts across millions of amino acids, to make this a bit simpler, we just threshold the activations at various different thresholds to create binary activations saying this feature is activated or not. And then we calculate classification metrics at each of these thresholds between the feature's activation and each of our concept annotations. However, one note here is that our features activate on much more granular concepts than most annotations are described by. So we kind of have to modify our metrics slightly to account for that. So specifically, here's an example where there's a feature that activates on one specific amino acid within histone kinases, And while this feature has perfect precision, meaning that whenever this feature is on, the concept is present, the recall, if you were to calculate it per amino acid, would be awful because it only identifies one out of hundreds of amino acids. And so instead, we have to slightly adjust our recall to say, if you activate anywhere within a given annotation, that's considered a true positive. And so when we adjust our metrics like this and calculate this between features and Swiss Pro concept annotations, we find thousands of features that activate on known biological concepts. 3 examples are here. And again, this is without the model ever explicitly being shown these annotations. So it has effectively identified all of these patterns on its own, which is pretty exciting. And another nice property of this quantitative labeling is that it gives us some measure of the interpretability of these features. So when we compare metrics for how well our SAE features match concept annotations, as the concept annotations in SwissProt, which is shown in pink, to the equivalent metrics, but actually calculated between the underlying protein language model neurons and the SwissProt concepts, as shown in blue. And then here we also actually add a baseline where the SAEs are trained on protein language model embeddings from a totally randomized protein language model, shown in green. So looking at the plot on the left, for each SwissProt concept, whether it's a zinc binding site or a beta sheet, we calculate the best F1 classification score for any feature to this concept. And this reveals that our SAE features have a much closer alignment with our concept annotations than the original neurons did. Visualized differently on the right, we measure the number of features, or the number of neurons, that are predictive enough of a concept to be considered labeled by that annotation, where we used an F1 score above 0.5 as our threshold for labeling. And we see that we can interpret up to 2,500 features per layer with an SAE, and only up to about 40 neurons per layer this way. And so this reveals that the SAE features identify many more known concepts and then also much more specifically identify those concepts than the original neurons do, implying that the original neurons contained a lot of kind of like interpretable information, but in a very polysemantic form. And yet the SAE was able to disentangle that into a much more interpretable representation. This is also what we see from just manually looking at the features and the neuron activation patterns. And the fact that none of the features from our randomized protein language model match any known concepts beyond some of them found individual amino acid entities, this kind of validates that this conceptual knowledge is actually being extracted from the embeddings themselves, and it isn't something learned independently by the SAE. And so we also find that, so most of this analysis discussed is going to be trained on the smallest variant of ESM, but when we scale this up to a larger ESM model, we see that again, even with a much larger embedding space, the neurons are still polysemantic. And also the SAE features trained on a larger protein language model actually capture way more concepts than a smaller one does, meaning that as we scale up our models, even though the polysemanticity remains, we can still actually extract increasingly more interesting information from these models. And so you might have noticed that there are way more features with concept annotations than there are actual concepts, which leads to the question of how all of these features connect together and how they relate. So thus far, we've only looked at the feature activation values. We can also look at the dictionary vectors for each feature, which, as a reminder, that's just the row in the decoder matrix for each feature, which just tells you when this feature is on, what is its direction in the original PLM embedding space. And so that gives you more of a sense for what is the role of this feature in the model. And so we can use these vectors to cluster all of the features. And when we do this, we see many features correspond to, or many of the clusters of features correspond to shared SwissProt concepts, implying that these clusters also capture many of the related kind of like functional features. And so if we zoom into some of the features corresponding to a cluster of kinase-related features, we see that while they each activate on kinases, and actually specifically they all activate on the catalytic loop of the kinase binding site, they actually have slightly shifted positions in terms of where within the kinase is most activated. And they all identify slightly different types of kinases. So the one on the left activates pretty specifically on serine-threonine kinases, whereas the one on the right activates on, in this case, a pseudokinase or other types of slightly atypical kinases. And then on the other hand, there are also many features that activate on similar conceptual concepts, but at different levels of specificity. So here we have 3 features that all clustered together based on the dictionary values, and all also were classified as TBDR beta barrels according to our SwissProt concepts. So if you look at the feature on the left, and if you actually examine all of the proteins that activate this feature, it's actually very specific to TBDR beta barrels in particular. And yet the middle and right features, if you look at the proteins that most activate them, they aren't actually specific to TBDR beta barrels. And here, they're singularly most activating proteins are beta barrels, but not TBDR beta barrels in particular. While they do activate on these proteins, they aren't specific to them. In fact, they actually activate on kind of beta barrels more broadly, including this particular variant. And so this kind of reveals slightly different levels of specificity in terms of how precise a feature is at identifying different subclasses of beta barrels, with the one on the left being most specific to TBDR beta barrels in particular. Now, the example also kind of points out a limitation in these SwissPro concept metrics, as not all biological concepts have binary annotations that can be classified. So, for example, there's no beta barrel feature in SwissPro. And so in this case, the more specific but slightly less accurate variant, which was the TBDR beta barrel, was applied to two features here, which leads to the question of, can we use a more unstructured method to help explain our features, particularly in the context of features that don't already have these like structured binary annotations? And so, as many problems do these days, this calls for a large language model. So to generate a description for a single feature, we select a bunch of proteins that activate our feature at varying levels, and then get a bunch of metadata on each of these proteins, both the binary annotations used earlier, and then also any unstructured text about the proteins. And we give all of this to an LLM and ask it to generate a description of what this feature is highlighting. And then we can validate these model-generated descriptions by asking a separate model to use only the description of this feature and then metadata about a bunch of different unseen proteins and try to predict the activation levels of these held-out proteins to see if this description alone is actually sufficiently informative about what this feature is capturing, that just based on the description, you can figure out which proteins would activate this feature to varying degrees. And so we can now evaluate how good a feature description is based on how well that description can predict held out feature activation values. So on the left here, we see that the majority of features are fairly interpretable this way, as measured by the correlation between the predicted and true activation values of our held out proteins. And then on the right, we see that surprisingly, the features that are LLM interpretable aren't necessarily the same features that were interpretable via comparison to our like binary tabular concept annotations. And there's only a correlation of 0.1 between the quality of the binary SwissPro interpretations and the quality of LLM interpretations, which on one hand I was very surprised by, because most of the information you're giving to these LLMs is metadata from existing annotation databases, but also just like looking through some of the features that didn't get good SwissPro annotations, sometimes you'd find examples where it activates on amino acids that are next to a specific structured concept, or maybe don't capture, like in the beta barrel example, they capture a clear interpretable concept, but it's not something that has an existing label. But it's cool that LLMs can actually pick up on some of those that the SwissPro annotations on their own, when measured in like a binary form, didn't get. So here we can see a few examples of feature activations along with their LLM descriptions and their true versus LLM predicted activation patterns. And we see that the kind of key identifying qualities of these features are indeed captured by the descriptions, which is super cool and makes it much easier to just like, as a person trying to interpret these models, makes it much easier to try to understand what all of these latent features are when the model gives you some kind of description about what you should be looking for. And so now that we have methods for extracting and understanding many of these features, we return to the original goal of trying to learn from the model. So one thing I discovered when looking through the SwissPro annotation predictions is that some features have almost perfect classification, where most of the time the feature activates above a certain threshold. It's on amino acids with a shared specific concept annotation. But then there are a few false positives where the feature is activated, but the annotation isn't present. And when you actually investigate some of these cases, I found that these false positives were actually picking up on examples of proteins that were simply missing the SwissPro labels. And through looking around at other online resources, you can actually confirm that these concepts really are present in the proteins. It's just the annotations aren't present in the databases. Which is super cool. It means that through these models picking up on these shared protein patterns on their own, they're able to find maybe more robust annotations for some of these structured concepts. And so this reveals a potentially new way to apply really specific existing annotations to new proteins. So for example, here, we can actually propose multiple new binding sites to a protein with no known binding site annotations, or potentially for features that don't even correspond to known annotations, but do activate on consistent concepts, it can help propose new annotations. And because these models learn this entirely on their own and it doesn't require any prior information, maybe we can identify patterns we wouldn't normally consider interesting or that exist in understudied or hard to study proteins, but proteins that are still easy to sequence. And so models can still train on them and learn interesting patterns. And then as another application, and honestly, more importantly, as validation of the features, we can synthetically activate features to guide or steer the model's output. To do this, we replace the intermediate latent embeddings for a given layer with the SAE reconstruction of those embeddings. However, we also clamp the activation of a specific feature to a really high value so that regardless of whether a given input activates that feature, the output will be affected as if the feature was on. And we can similarly turn a feature off that way. And then we just look at the new predictions when this feature is on and off and see how that feature impacts the model's outputs. However, the challenge with this lies in how to evaluate whether the steering is actually affecting the model in the way we'd expect given our interpretation. There are a lot of reasons why I think steering for protein language models and mask language models like ESM in particular is quite challenging, particularly compared to autoregressive natural language model steering. And this kind of results in two main failure modes when trying to evaluate whether steering a PLM feature actually has interpretable results. So on one hand, if you make the experiment too hard, you might just not get any useful readout. So for example, if you select a feature that activates on a complex structural concept, you might not actually be able to quantitatively evaluate the success or failure based on the steered sequence result alone. Or if the input sequences have too many masks or are too far from the intended kind of role of this feature, you might just be totally off distribution from the model's training and get a kind of nonsensical output. On the other hand, if you make things too easy, either in terms of the concept you've selected to steer or in terms of maybe let's say the starting sequence is too close to your intended output, there's a good chance you could have gotten the exact same intended results with a totally different interpretation of the feature, or even without steering the feature at all. And so what we did to get around that is so to kind of lab, we wanted to pick a case in which steering this feature would lead to a result that we only expected to see when this feature is on. So here we chose to steer 3 different features that all kind of similarly activate on this pattern of periodic glycine repeats that are particularly found in collagens. So this pattern is basically glycine, something, something, glycine, something, something, etc. And so we wanted to see if we turn on this feature, let's say we just turned on the feature on one of the glycines, you'd expect it to increase the probability of glycine. But again, that could also be explained by just saying this feature increases the probability of glycine. And so that doesn't really confirm this particular pattern. And so a bit more challengingly, we wanted to see if we turn on this feature at one position, will that impact the probability of glycine at the other periodic positions? Now, if this does exactly what we think it does, then ideally, if you steer one position, then every third position after that will also increase its probability of glycine, but none of the other positions will increase their probability of glycine. And so when we tried this in this particular sequence, indeed, that is the effect we observed. So just steering this first position in a sequence, well, we actually first have a start code on, but then if you steer the first glycine in the sequence, it has a kind of interpretable impact on the other positions, even the ones that are not directly manipulated by our model. And so in this case, you measure that by looking at the amount of how much steering we're doing influences the probability of glycine across different protein sequence positions. So here you see, as you steer this first glycine in the sequence, it increases the probability of glycine there, which is what you'd expect. But then also, 3 positions later, at the mask token, it also significantly increases the probability of glycine there. And as you try this with various different sequences, you also see the same results. You also can increase the length of this. And I think it was up to like 7 or 8 periodic repeats of glycines later, you still continue to see the probability of glycine only at the third positions increase, implying that this isn't just impacting this one position. The particular feature we found actually has a kind of interpretable impact on the other positions in the sequence too. And then if we rerun this experiment, but this time, instead of activating these periodic glycine features, we look at the 3 features that most specifically identify any and every glycine in the sequence, we don't see the same effect, implying that the signal we see can't just be explained by the model's knowledge about whether there's a glycine. So in this case, we're steering these periodic glycine patterns that are realistically not going to revolutionize protein design. And I'm not even yet convinced that PLM feature steering is, as useful as other methods for guided steering generation, but it's still a really interesting method for controlling the generation that does have the potential to guide sequence outputs and structure outputs without requiring any training on this particular property of interest. And also, maybe more importantly for now, it's a kind of useful feature validation technique and can confirm whether your interpretation of the feature actually is the impact of that feature on the model. I think down the line, there might be more interesting applications of this like protein feature steering. But yeah, for now, these are the only experiments we've done. And so as this type of mechanistic interpretability work is very visual, in order to understand any of these features, I had to just make a bunch of visualizations. And so originally just kind of made for myself, made this website that lets you explore all of the features in ESM, and you can see what activates a specific feature, or how your feature of choice highlights different amino acids in any protein of interest. And so we also released this website if you want to play around with the features. And in general, even though most of this analysis is like fairly quantitative, I find it really useful in terms of understanding what a feature does to be constantly like having a page of visualizers open and to be going back and forth between quantitative results and these visual explorations of what the features activate. And so our main takeaways from this so far are that the neurons in ESM-2 are indeed polysymmetric, as been observed in other modalities too. But with SAEs, we actually can extract consistent and interpretable features from the models. We can also make use of existing annotation databases to interpret these features, both by trying to classify our annotations by looking at binary annotations, and then also via LLM interpretation. And then we can kind of turn that around and use these features to help fill out missing annotations and potentially suggest new ones. And we can use these features to steer the model outputs in interpretable ways. And then finally, bringing us back to the original question of trying to use PLMs to discover new things about proteins. As I mentioned, we see that we can identify known biological concepts in new proteins. But looking forward, given that we can find many of the analyzed features have these, like they've effectively discovered known biological patterns without ever being shown them or taught about them during training, what do the rest of the features contain? Less than 20% of the features have these binary Swiss-print annotations. And so there's still kind of open questions about what else the models have learned that we haven't been able to explicitly interpret. And are they uninterpretable, or they're interpretable just with information we don't yet have well characterized? And so, yeah, what are the patterns that we haven't identified? And as we go beyond just identifying features, as Sniper's done, and we start figuring out how these features work together to actually make predictions within the model, what else can we learn? Now, on one hand, it sounds a bit ambitious and unrealistic to think we'll like extract new biological information from these models. But on the other hand, it also feels kind of unrealistic to assume that these models have only learned things we have already discovered, despite the fact that they're learning them in a totally different method and without any insight into human knowledge. So yeah, there's a lot of work to be done to thoroughly reverse engineer and actually extract information from these models. But I think we can learn a lot about biology in the process, learn a lot about the models themselves to help improve their capabilities. And yeah, I'm very excited to see what types of things we can actually learn, both with these models and kind of other biological models more broadly. So with that, I want to say thanks to my advisor, James O, for supporting me in exploring this area, for PhD funding from NSF, and then to all of you for listening. For any other details and analysis, you can check out the preprint. You can also play with the pretty protein pictures in the dashboard and use any of the open source code or models for your own exploration. And if you think any of this would be relevant to your work or have interesting application ideas, I would love to hear from you. I think people have already started using some of these models for various applications. So yeah, very excited to see how people with like specific domains of interest can actually find these to be useful. So yeah, happy to take any questions now. I think there are a few in the chat. Should I just read the questions in the chat now? Let's
Yeah, I
see.
think you can go ahead and if you want to read them and answer them, or I can read them and you can answer them.
Yeah, okay, so I'll just start at the top. Can linearly combining an SAE feature that may encode a functional motif into an existing sequence input to a PLM result in outputting a novel sequence with high confidence despite being far from anything in the training set may have had? Could this be used as a method to improve data augmentation and generalizability? Yeah, so basically the question is asking, can you use, wait, let me make sure I understood this. Okay, Fatima, let me know if I have misunderstood this. But so it sounds like this question is asking, can you take SAE features and use those to try to guide the model to generate new high confidence sequences that are totally far from things seen in the training set under the assumption that each individual component of this new sequence was something found in the training set? I think that probably is possible. It's unclear whether those will generate like functionally valid proteins, but would be a super interesting area to actually explore and try to validate. Are there ever instances where a single feature was found to encode for a diversity of secondary structures and domains that would otherwise seem unintuitive? If so, would you view this as a false positive? Yeah, so I think there are, of the features that we haven't like easily characterized through these binary annotations, I think they're a mixture of ones that are uninterpretable and ones that are interpretable, but just not necessarily intuitive. And it's not really clear right now, like what is the best way to properly differentiate between those two. And so like I've just mainly looked through a bunch of the features that we haven't interpreted yet. And for many of them, if I like look at them long enough and do research into those proteins, there is something interpretable about what they mean, or like about what activates them. But yeah, I think that's a really interesting question about would you actually consider these a false positive in terms of identifying features, or are these just things we haven't yet been able to interpret? Are the models freely available? Yes, they are. I think I haven't formatted them into an easy way, but I've just been like emailing to people who want them, but I'll put them on Hugging Face probably in a couple days because I've been meaning to do that for a while and just never got around to it. But yes, the code for training them is available, and I'll just put the model weights somewhere easier to download too soon. After training a sparse autoencoder, how do you define and locate a concept in an LLM in order to artificially activate it? So once you interpret what a feature is, so you train this sparse autoencoder and you get some number of features where that's kind of one of the hyperparameters of your model is how many features you get. So in my case, I have about 10,000 features, but have trained a model with varying numbers. So let's say you have these 10,000 features. The techniques I've used so far, either the kind of binary Swiss concept annotation or the LLM concept annotation, can help define patterns for these. And you could either identify a feature by saying, given a protein, what are the features that activate most highly on that protein? Or you can just scroll through the features that have annotations and say, find me a feature that activates on this particular type of motif. And then when you do that, you'll get the index of that feature within your sparse autoencoder. And then that is the specific dimension within your latent space that you clamp to artificially activate it. How do you decide how sparse to make the latent space? That's also a really great question for which I think there isn't like a super concrete answer within the field yet. So within the context of like language models and most mechanistic interpretability things I've seen, I don't think there's like a really principled way for selecting it. I think it's like a little bit based on the vibes of the features you get, because as I mentioned, you can kind of trade off how sparse it is and how good at reconstruction it is. And there's this like Pareto frontier of how sparse and how good your reconstruction is. And as long as you're along there, it's not really obvious what to choose. What I did in order to create like a consistent method for selecting across different proteins is I took a small subset of these sysport annotations and I ran that as an evaluation on all of the SAE models I trained within a given layer. And then I selected the model that had the most concept identified effectively. And so I used that consistently across all models to select the best model that have slightly different levels of sparsity. In general, these had like about between 1 to 200 features activated per amino acid. This like L0 metric of how many features activated per amino acid is like the general metric people use for quantifying sparsity. And so I use this kind of separate Swissprot eval of like known concepts to select my sparsity level, but it's not obvious that like that is the best metric that we care about to optimize. And I think like as people try to improve our models and also try to develop new applications, those might kind of have to go back and forth. And as you figure out what applications you care about, that might influence how sparse you want your model to be, or other hyperparameters about your model that will lead to the most useful features for your application of choice, which is also a bit specific to this like protein spaces. I think in like the language or image spaces, there aren't obvious applications to guide your choices about hyperparameters like this. Whereas here we like actually have potential practical applications that we can use to guide these choices, which is kind of cool. Do you plan on generalizing this work for interp of DNA, RNA language models? I am not currently doing that, but I do know that people are working on that. I don't think anything is out yet, but I kind of assume within the next few months, there definitely will be people, yeah, there'll be like some papers that have done that. And it's an area I'm very interested in, but currently not using that. How computationally burdensome was training the SAE? So the SAEs themselves, in this case, most of them are trained on the 8 million parameter version of ESM-2 that only has a 320 hidden latent space. And then the ones I put in the paper only expand up to 10,000 features. You can train ones that are like have a larger dictionary of latent features than that. And so the training of those wasn't actually the most computationally difficult step. Given academia, I like don't have the most compute available. And so it was like a little bit slow, but not nearly as annoying as just getting the features themselves from the ESM models, because those are way more computationally complex and take way more time than running the SAEs. And then also once you've trained them, actually like doing all the analysis on the features themselves is more computationally burdensome than training the SAEs in the first place. So yeah, I think those ends up being more of a limiting factor. If you worked with a smaller subset of proteins with common structural properties, would it be easier to identify what features map to what? I'm not sure this means in terms of, would this be easier for the SAE to identify those features or easier for me as a human? Feel free to clarify that. I'll just kind of answer one of those. But so from the perspective of SAE, so people have actually found that if you train your SAE on like a specific subdomain, so in this case, like if I trained my SAE exclusively on proteins within a certain family, it's quite likely that the types of features you'll extract will be much more nuanced within the context of that family. And so it'd be much easier for the model to extract out relevant properties related to these proteins if they share some kind of structural or functional properties. But then, of course, your SAE wouldn't generalize to other things. And then in terms of me, like as a person trying to analyze them, I guess it would be easier if you like had a more well-constrained domain and then you could identify features that are really specific to that family. So like, for example, with the Swissprot binary annotations, within the concept of protein kinases, there's just one feature mostly that's, or there's like one concept that says like is a kinase and will span hundreds of amino acids. And if I was really focused on just kinases in particular, I might use something like Cliffs that has way more nuanced information about what each amino acid within a kinase or kinase binding pocket is doing. And then it'd be easier to find more nuanced features about those there. But then again, that only really generalize to that particular family of proteins. How to select features according to established concepts. Like if I want to look at a flexible loop part of proteins. So in this case, there definitely are concepts like one of the concepts within Swissprot is this a disordered region? It doesn't have the level of specificity of is a flexible loop, but you could potentially like combine existing annotations to get that. Or really, if you want to create any type of annotation database yourself, or if you have any other method for defining some concrete table of, for each amino acid in a protein, annotations of is your concept present, is your not concept present, then you could totally use that to evaluate which features correspond to your personally defined concepts of interest. I just chose these because they were kind of comprehensive, but it's very easy to generalize this to any other type of binary like amino acid level annotations. Or you could like take a protein level annotation and spread it across the amino acids. Um, was there any reason to choose ESM2 over the other PLMs like Pro-T5? Would this be consistent with other PLMs too? Honestly, it's just been really well studied, and like people have found specific patterns in the attention patterns already, and there's been some work on it, but it was a pretty arbitrary choice and could just as easily be applied to other protein language models. I definitely in the beginning was kind of interested in more autoaggressive protein language models as opposed to masking-based protein language models, because I think steering is a little bit easier when the models are more explicitly trained to just like go off and generate a bunch of sequences. And I think the masking made it much more harder here. But it would be really interesting to see if the types of representations you learn with those are consistent too. I'm not as familiar with the literature, but I know there has been some work trying to do like look at the attention patterns within protein language models with other types of architectures, and you find slightly different results. And so it'd be really interesting to see, how much do these architectural decisions and the training structure, how much do those influence the types of features learned by the protein language models, or are there just like certain universal features that are broadly identified across protein models? So if anyone's interested, that would be very interesting to explore. Do the types of features that are reliably identified or fail to be identified inform what kinds of downstream function prediction tasks they may be best suited to? So I haven't evaluated that, but I think that'd be really interesting to answer. I kind of assume that would be the case, that like if there's certain types of features that you don't like, to the question that was just asked about how this differs between models, it'd be interesting to see if like you can find an evaluation where one model is much better at a certain type of task, the other is not, and that can kind of be explained by the features that are or aren't present. I looked into that a little bit between the smaller ESM and the larger ESM model, and there were certain types of concepts that were identified in the larger ESM model, but not the smaller ESM model. But I haven't yet like found good external evaluations telling me whether those concepts are correlated with the types of tasks that the model gets better at with scale or not. I kind of assume that would be the case, but haven't evaluated that, and it'd be cool to see if someone could like create a kind of like robust evaluation for whether that's the case, because it could be a super interesting way to, yeah, to one of the early points, like actually help improve model development. Also, sorry if I've missed any of the questions. I'm trying to go through all of them. I think I've got most of them. Sometimes input information can die out at different layers or depths of a LLM. How did you choose which layers to grab? And do you have multiple SAEs corresponding to different lengths? Can dictionary size value by depth? Yes, so I actually just trained on every layer of the small ESM, because ESM, the 8 million parameter model, only has 6 layers. So I just did all of them. And I kind of like went through everything a little bit quickly because there's a lot of results. But if you actually look at the figures, the very first layer, the features you find there are like very different from what you find in all the other layers. And that layer's already gone through like one round of attention, but you mostly find features that correspond to specific amino acids or like kind of like really small local properties. And then as you go further in the model, you start to see way more complex properties emerge. Yeah. And then for the larger model, I just like picked kind of evenly spaced ones. And then can dictionary size vary by depth? Yes, you could totally train, in this case, just for consistency. The ones that we put out and analyze most all have the same fixed dictionary size, but in theory, you could totally vary the dictionary size by depth. It was a little hard to compare the types of features you get in some layers versus another, because they are technically all different SAEs that have slightly different hyperparameters to them. And so some of the variance is just due to like kind of small retraining noise or different hyperparameters. But there are more recent methods like crosscoders that are kind of specifically targeted towards trying to compare different layers of a fixed model to each other in like a more consistent way. And so that can maybe be better at answering this question of what varies between the different layers. Okay, cool. I think those were all of the questions. And if there's anything else, feel free to email me. Happy to, yeah, answer more.
Thank you so much for your presentation. It was awesome, and thank you guys for all of the questions. Um, yeah, if there aren't any more questions, I think we can hop off. Thank you again.
Oh, yeah, thanks so much.